Good morning, everyone. Thank you for joining. We will wait for a couple of minutes for other participants also to join. So I request you to stay in the meeting. Thank you.
Recording.
Good morning, everyone. We are happy to see you yet another with another webinar. Thanks for joining. Giving you a brief introduction, myself, Annie, I'm the senior application scientist at Christian Biosystems. And giving a short brief on what we do, we are established in 2003, which is a biotech firm based in Mumbai. We focus on understanding our customers' projects from a technical standpoint to provide cutting edge solutions to scientists to support their research. We have a strong background and expertise in immunology and working with biopharm, CROs, vaccines, and academia at pan-India level. Uh, Talking about the ABM Group, which was founded in 2002, is based in Vancouver, Canada. They expertise in developing and commercializing novel research tools like PCR reagents, vectors, viruses, and a complete portfolio of unique services have made them an invaluable partner to laboratories and research institutions around the world. So today we have Ms. Heidi Chu with us today to give us an insight on the gene delivery revolution, exploring the latest advances in AAV lentivirus and adenovirus vectors. So talking about Ms. Heidi Chu, she is the product marketing manager at ABM, bringing a solid foundation in both business and science with a joint degree in business and molecular biology from BCIT and the University of British Columbia. With over 8 years of experience, Heidi possesses deep expertise in viral vectors from packaging and production to advanced applications in cell engineering, including cell immortalization. She also played a key role in establishing high titer AAV purification methods at ABM, and Heidi's in depth in the understanding of viral vector applications, paired with her strategic marketing insight, strengthens product positioning and go to market strategies across Northern American, European, and Asian markets. She also collaborates closely with teams on product education and support, driving brand impact and enhancing customer satisfaction. I would request everyone to be on mute. In case while she is running through the presentation, if you have any questions, you can drop it in the chat box, which will be addressed in our during our Q&A session. And now I would like to hand over this further session to Ms. Haley to continue with the presentation. Thank you.
Okay. Thank you, Annie, for that wonderful introduction. Hello, everyone, and thank you for joining. Like Annie said, my name is Heidi. I'm the product manager here at Applied Biological Materials. I'm going to go ahead and share my screen. And off we go with the presentation. Okay. So today we're going to be talking about emerging technologies in gene delivery, specifically with advancements in AAV, lentiviruses, and adenoviruses. Now, sorry, I think there's some background noises. Is it okay if I ask everybody to be on mute? Because that would also sort of cut into, it switches my screen. Thank you so much. Okay. So the summary of topics that we're covering today, so obviously highlighting the importance of viral vector in cell and gene therapy, some of the key challenges that we have been seeing in the past decade and what have been done to resolve these key challenges in terms of advancements. And of course, in addition to the advancements that we have made, what other tools are there that we can use to enhance any efficiency, efficacy, and increase the viral vectors in therapeutic potentials. Lastly, we'll talk a little bit about scalability and manufacturing, some real-world examples that you will find throughout the presentation as well. And lastly, the takeaway messages in case you're looking to do some preclinical research that will lead to translational cell and gene therapy in the future. So I know Christian, Annie has done a very good introduction about ABM. Just to kind of add a little bit on top of that, ABM is founded in 2005. And we're headquarters in Vancouver, Canada. We have branches in China, France, and US. And of course, we work with over 60 distributors worldwide, including wonderful partners like Christian, to deliver our products into the different territories. Now, we work with many academia as well as pharmaceutical companies, helping them with viral vector designs, delivery, scale ups, as well as cell engineering. And really the driving force behind our success is our founder. So in this talk in particular, I want to mention our founder because really he is the driving force and he is the scientist turned entrepreneur. He has very strong foundation of recombinant technology just from his experience as a WHO fellowship in UCLA. And he has also done many researches and spearheading the world of viral vectors in terms of adenoviruses. So he actually helped develop the cloning techniques and amplification techniques at CloneTech when he was in the Bay Area. And of course, that kind of translates into the cell engineering techniques when he founded ABM. So as we all know, cell and gene therapy are reshaping the biopharmaceutical company. And today there are at least 22 cell and gene therapy approvals with FDA. And there's many more ongoing clinical trials that's targeting a diversity of diseases and conditions. A lot of them are in cancer. There's many that are targeting inherited diseases, infectious diseases, and you name it. And really, what is the driving force behind cell and gene therapy? A lot of it is the viral vectors. So they are the basic process of gene therapy. So viruses, as we all know, they're very powerful. They have evolved a way of encapsulating and delivering their genes into human cells, right? We know it in a pathogenic manner. Now, scientists have tried and successfully harnessed the ability to manipulate the viral genome to remove the disease-causing genes and insert therapeutic ones. So one of the classic ones that everybody always talk about is the lentiviruses. It is one of the most well-known recombinant viral vectors, and it always amazes me at how people were able to, come up with the brilliant idea of just keeping the key components of the viruses, right? So what it takes for a virus to enter a cell, right? You need that encapsulating ability. You need that capsid to make sure that it amplifies. You need to make sure that it ensures transcription, that inserts into genome. So recombinant viral vectors is really taking into what is essential for that virus to become a virus and gutting out anything that we don't need. And scientists, in addition, put the gene of interest into that. So it's really we're leveraging the pathway of these viral vectors and its ability into infecting cells to our advantage. So lentiviruses is one of the most well-known ones. There are many different other ones, and really they differ in how well they would transfer the genes into cells that they will recognize and how are they able to infect and the mechanism. So whether or not they're altering the DNA permanently or temporarily. So this chart here kind of shows many of most common viruses that could be used in therapeutic potential. There's pros and cons of each one. And of course, that comes with the challenges and the advancements on how to solve that. So lentiviruses, for example, is one of the most well-known ones. The packaging capacity refers to how much of the gene size you can insert. And so when you think about it in a therapeutic potential, if you're trying to do translational, into clinical, trying to cure something, say if an inherited disease is lacking a gene, an enzyme that's causing a disease, you will have to insert that into the cells so that it will express the gene so that you will cure the diseases. Now, with lentiviruses, you can only insert to a capacity of 9 kbg. And actually, that's on the bigger side because when we look at AAV over here, that's only 4.5, right? So when we start thinking about what viral factors to use in the research that will lead to translational, these are kind of things that we kind of think about. what we're trying to do, what is the size capacity, and does that fit into the specific viral factors that you're trying to use? Another good consideration that we need to keep in mind is the transduction efficiency. So not all viruses have the same virulence, we would say, right? So if we think about, flu, COVID, every single one has a different sort of infection efficiency, and it's the same for these recombinant viruses as well. So adenoviruses, for example, has the best transduction efficiency. um, every single one has different targets as well. So I like to use COVID when I talk because everybody knows about the COVID. It targets, airway cells, right? So lentiviruses, for example, they target most dividing cells and non-dividing cells. Adenoviruses, they do the same, but actually it has a higher transduction rate towards specific cells like primary cells, right? So these are things that you will have to keep in mind when you're trying to develop whatever you're doing in terms of therapeutic uses, applications down in the future. AAVs is very interesting because naturally occurring AAVs have very specific tropism towards specific cell types. So even though it says all cell types, but different serotypes of AAV actually targets different tissues. And scientists can leverage that and use specific AAV serotypes to target specific diseases. So I do have a table in the later half of presentation that kind of shows that. So it's just kind of good to keep in mind that, again, what is the goal we're trying to achieve here, and what viruses you could use that to your advantage. Another good consideration is the expression. And this one really points out to the downside of lentiviruses. Lentiviruses are known to do stable integration. So it's good when you're looking at basic research, you're trying to figure out something, stable expression is good, but it's not so good for therapeutics, right? So thinking about treating humans, it raises ethical concerns as well as regulatory issues. We don't want anything that is permanently disrupting our gene. It causes a safety issue. So lentiviruses, that is the key challenges that we're facing right now. Adenoviruses, it has a very high immune response. And so one of the past cases actually was a previous FDA-approved drug that was using adenoviruses, and it actually causes too strong of an immune response in a patient that it causes the patient to die, right? So that is the challenge that we're facing with adenoviruses if we're thinking of using it in potential therapeutics use. AAVs, sorry, my voice is a little cracking because I'm a little sick today. Hopefully that's okay for everyone still. AAVs, like I was saying, it has very specific tissue tropism, so it kind of limits what we can use it for, right? So natural AAVs, we have AAV2, we have AAV4, 5, 6, and that only limits to infections, in the central nervous system, in the liver tissues, in the skeletal muscle tissues. But what if we want to explore outside of these tissues? So that is the limitation for AAV. It kind of has too little serotypes that we will have to make modifications to these serotypes so that we can increase the usability in therapeutics. So kind of to summarize the key challenges here we're facing, lentiviruses, like I was saying, safety is a huge concern. It integrates into the genome, it will make permanent changes, and that raises regulatory and ethical concerns. Adenoviruses, on the other hand, has a very strong immunogenicity. And so it affects safety, it causes somebody to pass away in a previous clinical trial, and that was one of the key challenges we're facing. AAV has limited tissue tropism, and so we need to broaden that tropism in order to deliver to previously inaccessible tissues. So some of the key advancements kind of addresses these drawbacks as well. So when we talk about safety for lentiviruses, what scientists have done is that they have made the next generation factors that it's called self-inactivating. So these are the IDLVs, and that has the improved safety profile. For adenoviruses, we need to reduce the immunogenicity in order to increase that therapeutic potential. And the way to do that is find different adenoviruses out there, different types, maybe from monkeys, so that we can start using that into therapeutics. And AAV, we would be expanding the capsids. This is the serotypes in order to improve the tissue-specific targeting. So specifically how each one is done, the IDLVs, what was done is that we mutated an integrase enzyme in order to prevent that integration to the host genome. So what that means is that when you use this IDLV lentivirus in your experiments or later on in translational, the factor actually forms episomal circles in the host cells. So it actually stays as a transient expression instead of integrating. So that takes away the safety concern. And so this is particularly advantageous for therapeutic applications because there's no permanent genetic changes. And they would be very suitable for transient expressions where it's sufficient, such as delivering immunomodulatory genes or in activating repair pathways. The ways to reduce immunogenicity in adenoviruses would be using different serotypes. So most commonly used is adenotype 5, but now there has been research into adenovirus 26, adenovirus 35, or non-human adenoviruses. And so these modified capsid proteins will reduce the immune recognition in humans while maintaining the efficiency. So that's a very new and interest pathway that people are taking to look at adenoviruses because it does retain its advantages of broad ability to do transduction. But on the flip side, a lot of people are thinking strong immunogenicity can be leveraged, right? Especially in vaccine development. So in vaccine development, you actually want strong immunogenicity because you're eliciting the immune response generating antibody so that it can actually fight off infection. So again, I think when we talk about what viral factor to use in terms of therapeutics down the road, we have to think about what is the end goal. So you can't really just look at, adenoviruses, I don't want to use that because it's too immunogenic. But if you're doing vaccine development, that is something that you want to use. So I think that is a common thing that I will be talking about throughout the presentation. It's really, you have to have a clear goal in mind on what you're using the viral factors for, what is the end goal, and what do you need in terms to getting yourself to that therapeutic application in downstream. Coming back to the AAVs, they were limited by natural serotypes. So what happens is that scientists have been using new hybrid capsids, and these are created through direct evolution. So what that means is we do a generation of libraries of AAV capsids, and then we would screen them for desired properties. So as an example, a serotype called AAV-DJ came about by combining sequences from 8 different AAV serotypes. So 2, 4, 5, 8, 9, and others were combined together, and this creates a novel serotype. And we screened that, and we found that it has enhanced properties, including a broader tropism, and it also improves cellular uptake as well as reduced immune recognition. And kind of going further with that, AAV-DJ8 is another revolution of that. So it does retain features of AAV-DJ, but it has an enhanced ability to cross the blood-brain barrier. So with more iterations of these evolutions, there's more AAV capsids that is coming out as a result of the researchers around the world to improve the tropism of AAV. So the 3 that we have been talking about actually works on the viral factor itself, but they're actually tools that will enhance the efficiency and therapeutic potential. So one of the most well-known ones that has taken the world by storm is the CRISPR-Cas9 based editing to enhance precision targeting in any platform, right? So you can still use CRISPR-Cas9 with a lentivirus if you want to knock out a specific disease-causing gene in a inherited disease, or you could use CRISPR-Cas9 in a HDR pathway if you're looking for repair-specific inherited disease, such as sickle cell anemia, by repairing one of the genes that's faulty. Another way that we can look at enhancing therapeutic potential is actually changing the vector design, right? So instead of looking at how do we change the viral vector itself, the design, you can optimize promoters that can potentially give you a higher specificity in terms of tissue expression and to have better control of the gene. And lastly, we can also make some service modifications to, again, enhance that cell specificity. And one of the best examples I would say would be the CAR-T therapeutics that people are looking at. So CAR-T uses lentiviruses, and what it does is that it takes T cells from human patients and inserts in the chimeric antigen receptor, and the receptor has an extracellular domain, it has a transmembrane domain, and it has a signal domain that is presented on the T cell, right? So these are surface modifications on the T cells that will help direct these T cells when it goes back in the patient through infusion to target specific cancers, right? So one of the best examples would be a CAR T cells that expresses CD19, chimeric antigen receptor that is used to target and kill B cell cancers. So just kind of diving into specific, the ones that I talked about, CRISPR-Cas9, I'm pretty sure a lot of people know it already, but I like to think of it as a knife and a hand. So Cas9 being the enzyme would be the knife, and you always have to have a sgRNA, which is a guiding hand, and it is one of the revolutionary gene editing, precision gene editing examples, right? So once you have a hand and a guiding knife, you can cut anywhere you want. So this is talking about knocking out a gene. You can knock in a gene. You can also use the cellular pathways, HDR, to repair anything you would like as very specific locus. And it's much more than that, right? So when we talk about CRISPR-Cas9, there's many other variants that we can use. And there's effector domains with the dead Cas9. There's also base editing. So you can turn on the gene, you can repress the gene. Again, I think once you know an underlying causes of a disease that you're trying to cure for, then you would start thinking about, what do I need to make that happen? Would it be using CRISPR-Cas9 to get to a specific location that I need to turn on the gene so that it reverses the disease phenotype? Or would it be using CRISPR-Cas9 doing demethylation or methylation, right? So as long as you know what is the mechanism of the disease that you're trying to look at, what is the therapy that you're trying to cure for, there's a lot of things that we could do with CRISPR-Cas9, again, in combination with the viral vector. So CRISPR-Cas9 could be used in combination of lentiviruses, with AAV, with adenoviruses. And that question kind of goes back to, what is cell type you're trying to use? What is the target? What is the key tissues that you're looking for? Maybe you want to use the AAV. So all of that kind of comes together when you're trying to design a vector virus for that. Another way to do the optimization would be optimizing the promoters. So like I was saying, AAV, if that's the vehicle that you're planning to use, it already has a tissue-specific tropism, but you can also further enhance that. Lentiviruses doesn't have that. So one could use promoters, specific tissue promoters, to drive that expression in that specific region. So an example here that we have is that we made a lentiviral expression vector expressing sepnasin-1 promoter, and this promoter specifically expresses in neurons, and they were able to see specific neuron gene expression. So you could use specific promoters to kind of get what you need to do. And at ABM, we are able to help you with editing, cloning, gene synthesis. We do different promoters. And if you're doing earlier part of the research, you can look at tagging with different GFPR failure surface to kind of help you evaluate the progress before you kind of put that into translational. One of the last ones is surface modification. So I already talked about a classic example of lentiviruses and CAR-T, the chimeric antigen receptor. But of course, AAV serotypes is another classic example of how you would alter a surface modification so that you can target specifically the tissues that you want to do. So here are just some examples of the ones that is already available. But of course, like I was saying, a lot of research is going on with broadening the serotypes. So there are more that is going to be coming out. So one way I always get asked is, what is the best serotype to choose in my application? And I would say, honestly, a lot of them kind of cross different specific tissues. But the best bet is, looking at current developments, current research that's going on there, and see what other colleagues are doing in terms of what serotypes they're using. And you can consult, what's being FDA approved that they're using, and you would kind of go from there as a starting point. And of course, if you have any questions, we're always happy to help. We can help you look into literature to see which one is the best suited for you. But that is probably the way to go. And once you kind of figure out, what you would need, you would do some preliminary studies, and it always comes to a challenge for scalability and manufacturing challenges. So what we would always address is that, when you're working with a vendor, you're always looking at the quality, the purity, and the yield. In terms of quality, how we know it's qualified, of course, you need to ask to the validated sequences. So a good vendor will always show you that whatever vector they're using to package the viruses are expressing the sequence that you need. And of course, in terms of purity, there's different purification methods, and depending on the yield, there could be skim columns. For AAV, it could be allotexinol. So it's always kind of good to check with the vendor on which purification method that they're using and whether it fits your protocol. And purity is very important, especially when you move into animal models, right? So if you have any residual salts, residual DNA that is interfering, it can interfere with your experiments. In terms of yield, one of the quality controls that we always do is titering, right? So we titer for physical titer. So what that means is not just looking at we actually do physical and functional titer. So physical titer is just looking at the number of capsid in the viruses, whereas functional titer actually counts the active viruses. So when we do qPCR titering our lentiviruses, for example, we actually calibrate that into functional titer, meaning we would cross-reference with zero dilutions of GFP lentiviruses and kind of count that functional titer, cross collaborate into a qPCR to get that final titer for the customer. And of course, in terms of quality control, it's always good to make sure that mycoplasma is being tested throughout the entire production process, because that could also interfere with the results and the actual active viruses that you would have. When it comes to scale up, a lot of vendors will have to move from adherent plates into bioreactors. So these are roller bottles. So a lot of adaptations will have to be optimized. So some of the examples would be if you move the cells into roller bottles, obviously it's not adherent cells anymore, it would be suspension cells. And so that adaptation is required, of course, the media. And when we look at bioreactors and eventually moving into pharmaceutical therapeutics, it's very important that, we're not introducing any FBS, any other animal products that is not accounted for. So a lot of times when you talk about bioreactors, you have to make sure that the media has been switched to sugar-free and that it is able to sustain and to keep that high titer as well. And of course, when you scale up, you have more value, then it comes down to the purification method. So it's very important for a vendor to be able to have access to ultra centrifugation, for example, big segment columns to do that scale up work. So what I'm trying to say is that the production ready facility is very important when you're looking at manufacturing challenges. And a lot of times you do have to work with a CRO specifically to do that scale up. So one of the examples that we have worked with is the Parkinson's disease. So the idea from our client is that they're trying to introduce a gene that would produce GABA, right? So when we talk about Parkinson's disease, the main overactive brain region is the subthalamic nucleus. That would be introduced with a gene that would produce GABA, and this is an inhibitory chemical. And so once you have that inhibitory chemical, it could potentially quiet the brain region that and affiliated tremors. And so what we have done is that we have made an AAV2 for them, and with local anesthesia, the inactive AAV2 actually delivers the GAT gene, which produces GABA into the patient's subthalamic nucleus, and the gene will instruct cells to begin making GABA so that it reestablishes normal chemical balance. So this is all in the pre-clinical stage. Of course, afterwards, they were able to find a GMP facility that were able to support the clinical trials, but we were involved in that preclinical stage in developing the vectors for the client. So the main takeaways is that, viral vectors actually plays a key role in future cell and gene therapy landscape. It's always evolving. There's always going to be new viral vectors that is in place. There are some key challenges, but it has always been overcome with new technologies such as CRISPR in terms of improving precision or vector design, like the promoter, to help with specificity, efficiency, efficacy. So understanding what your end goal is actually very crucial in selecting your approach. And this is kind of like where ABM equation talk comes in, right? So I think throughout the presentation, we've been talking about it's very important to keep an idea on what is your end goal and what model you're trying to use, what you're trying to deliver. Are we talking about looking at a final therapeutic that's going to be working on dividing cells, non-dividing cells? And at the current stage, are you looking for in vitro preclinical, or are you moving into in vivo? So those are kind of the things that you have to think about, right? So, and what type of expression are you looking at? Are you a stable expression good for you? Or do you need transient? Are you looking at knockouts? Will you be using siRNA? Will you be using CRISPR? So all these questions should be questions for you to think about. And of course, we talk about insert size because across the different viral vectors, the lenti, the adeno, the AAV have different insert size limits. And of course, when you're thinking about preclinical, maybe if into clinical, what kind of reporters will you need? Right? So, and in preclinical, when you do in vitro, perhaps you want to do proof of concept, then it becomes important how you're going to make that proof of concept. Do you need some markers in place? Do you need a GLP pyramidin to kind of help support your model and make sure that it works? So all these consideration is something that you need to think about. And lastly, I know we talk about viruses a lot, but you can also deliver it in plasmid format, right? So plasmid format can be delivered in electroporation. I know people have done nanoparticles, delivering specific genes. People have delivered microRNA using LNPs, right? So all these kind of has to come together, and we're happy to start a conversation in terms of what you need and help you kind of guide you through what you need to do for your viral factors. And this is kind of like a brief overview for what we could do. So I know we touched upon gene therapy. We talked a little bit about CRISPR. We talked a little bit about RF, right? So if you're talking about a disease that you're trying to look at that is lacking a specific expression, like the GABA in Parkinson's disease, that would be the RF. There's some gene therapies that is actually leveraging the microRNA, where it targets the 3' untranslated regions to upregulate or downregulate a gene. And of course, you can do some knockdown siRNAs in gene therapy as well. So all these applications can be delivered in terms of lentivirus, AAV, adeno, and retrovirus. Again, it depends on your end goal, what you're doing. And of course, at ABM, we are able to support gene editing expression, gene expression studies, vaccine development, or regenerative medicine as well. So what makes, I'm just going to end this off with what makes ABM different. So hopefully I have convinced you that we do have the expertise in terms of vector viruses, what they do and how we can do it in terms of cloning, what kind of angles you have in place. So we do have that guidance for every step along the way. We are able to do personalized solutions for your unique needs, and that we offer a one-stop solution for your research project. So I would say if you have any questions, talk to Christian, talk to us, and we are happy to start a personal conversation one-on-one with you in terms of what you need in terms for vector virus and maybe your therapeutic applications down the road. And with that, I would like to take the questions and answers and kind of end from here. So hopefully it was kind of clear in terms of what you will need to do. Really, the drive home point is that for everything that you do, you have to think about the bigger picture, right? So you're thinking about what vector virus to use, which one to pick, what is the target, what application I'm going to be using, what implication does that mean, and what kind of consideration I need to do in terms of safety, in terms of immunogenicity, and in terms of the broader serotypes and tropisms that you're trying to target.
Okay, thank you for your presentation, Hedy. So we have a couple of questions to ask to you. So the first one is, are there specific advancements or techniques you believe will define the next phase of gene delivery development?
I think the one that I think was the most important would be anything along with the CRISPR line, right? So I know we didn't talk specifically about everything that CRISPR could do, but there's so much more you can do with CRISPR. A lot of people say it's knocking out only, you can do some knock-in, right? So if we're talking about a gene mutation that's caused the disease, what one could do is they could use CRISPR. They could do gene mutation knock-ins to fix that mutation so that it becomes a normal, right? So that's one application. So I think that is very powerful. We have experienced CRISPR hands-on, right? So we know how easy it is. We have experience with previous gene editing tools, such as Talon, such as zinc fingers, and all of these take years to get that precision to take each design, and nothing is easier than CRISPR. So I think combining the technology for CRISPR with viral vectors, and of course, our ability to do ex vivo work. So, for example, taking stem cells, taking germ cells, taking embryo cells, and make that modification, put that into patients, and that brings a lot of potential.
Okay, thank you. One more to go. So, how do the approaches to vector development differ from industry and academic settings, and what each can learn from the other?
Can you repeat that question again? Sorry.
How do the approaches to vector development differ between the industry and the academic settings, and what each can learn from the other?
Right. So I think there's so much things that we can learn from academics. So basically, a lot of academic settings that what they do can be translated into translational research, right? So when I was talking about the serotypes for AAV. So that came from academia, right? So they were able to use massive screening computational approaches to increase that tropism. And that kind of translates into industry, right? That helps industry and say, hey, now there's a new serotype that you're able to use for blood-brain barrier, right? So I see there's a lot of back and forth between academia, industry, and one feeds into another, right? So now industry kind of says, oh, I'm looking at this cancer cells and I need another cell type for AAV that can target this specific tissue. Are you able to do that? So it's a crosstalk, right? So they kind of send that feedback into academia. Academia is able to use their computational powers to generate another cell type that will feed into the industry. So you can't survive without one another. So it's very interconnected.
Okay, okay. Thank you. So I couldn't see any questions more. So thank you all for attending our webinar today. We are grateful for your participation and hope you found the session informative and engaging. We appreciate you taking the time out of your busy schedules to join us. Your encouragement and the questions contributed greatly to the success of this webinar. A special thank you to Ms. Heidi Chu for sharing her expertise and insights on the latest advancements in the AAV, lentivirus, and adenovirus. The recorded session will be sent to you, and keep an eye out for our email on promotional offers only for the registered participants. Thank you once again. Thank you for joining. Thank you, Heidi.
Thank you. Thank you, everyone. Have a great day.
Thank you. Yeah, same to you.